Hello friends, today All Choice Education has brought you the almost very selected questions of principle and biotechnology, principle and process. Friends, this chapter requires a very good concept. ये concept based chapter है. अगर हम एक बार इसके चैप्टर्स के सारे कॉन्सेप्ट को पता कर लेते हैं तो बस हमको इस चैप्टर में ज़्यादा क्वेश्चन कहीं से भी दे दे it will help you to click the right question. But today I have brought you very selected 30 questions and let us practice and try to secure the 100% mark. Friends, question is like that is it is started from its definitions. That is according to EFB. What is the EFB? What is EFB? It is that European Federation of Biotechnology. He has given a definition that the integration of the natural science and the organism, cell, part thereof and the molecular analogues of the product and service is known as. Means he has defined the biotechnology, that how it can be defined. So here we will use that the integration of the natural science, whatever the nature tells us, and about the organisms. Any part, cell part, means cell is the basic unit of the life. So cell. So all this which is known as in the service, the product in the service is known as what? Biotechnology. I haven't make you the syllabus, the option like A, B, C, and D, because directly we will come to the answer, and it will very easy for us to remember the part. Next question is that Agrobacterium tumefacin used in the genetic engineering for means the bacteria which is used, the Agrobacterium and tumefacin in the genetic bacteria in the which mode? So it is in the vector mode. Next is the biological function gene coding for the tyrosine tRNA, that is translate or transfer RNA of the E. coli synthesized by the Khurana, is this Indian based scientist in 1979, had how many nucleotides? That is what is that? 207 nucleotide pairs it was having. Then it comes here to who isolated the first restriction endonuclease. So who there is a two type of restriction enzyme, endonuclease and exonuclease. So here endonuclease restriction enzyme who has discovered, that is the it is discovered by the Nathan and Smith. Next question, friends. Next question is that we are having the thermal cycle. Thermal means hot. Thermal means the things which is hot. Thermal cycle take place in which techniques? So there are the various techniques we will coming across. I will make this separate questionnaire for the techniques. So this is the introductory that the thermal cycle take place in the which techniques. So it is in the PCR. What is the full form of the PCR? Polymerase chain reaction. Is a polymerase chain reaction. Okay. So there are the various steps. So here that is the thermal cycle, means it is here 95 you will find, 75 you will find, 45-degree find, and many the bacteria or the things you will use which is working in the heat. So that is why here it is written the thermal cycle. Take place in which techniques? So don't get confused that it is gel electrophoresis or anything, that is the PCR. Now here in rDNA technology, R means recombinant DNA technology. Which of the following techniques is not used? Here see, not used. Sometimes question is asking used, sometimes it is not used, sometimes it is asked which is the pair, sometimes it is asked which is not the pair. So here it is asked that in rDNA technology, that is recombinant DNA technology, which of the following technique is not used in introducing DNA into the host cell. So when we introduce DNA into host cell, the various technique we use like micro injection, okay, and heat and shock method. So here that which is not used, that is not used a conjugation. Conjugation is not used. Then what is the PCR? PCR technique is used in. Now here the same thing, PCR technique is used, the polymerase chain reaction is this thermal cycle. Where it is used? This technique is used for what? For the production of genetically modified food. Okay, it is also used. Now here, the next question, what you can see in your scheme is the BACs and YACs are. What are these? So these are the DNA what we use from the bacteria and yeast. Means here artificial DNA obtained from the bacteria and yeast, that is bacterial, artificial and yeast artificial group. So they are used accordingly. So like this they have got their names also. I hope you are getting. Next question, let us see. When the genotype of organism is improved by the addition of foreign gene. What is that? When the genotype of an organism. We know that phenotype, what we see, what is appearing, that is the phenotype. And the genotype is what? The genotype which makes the phenotype to be appear. So it is the setting of the gene. So that is when the genotype of organism is improved. Improved means that is manipulated. So by addition of the foreign gene, when we add, see, suppose one DNA is here, then other DNA is added over here, the DNA is added here, so this is the foreign gene, this is the foreign gene, and this is the host. So which technique it is known as? What is this? The process known as what? Genetic engineering. So we are doing in the genetic engineering, we are doing the same thing. What we are doing? We are taking the foreign gene, we are ligating them, and these all things we are doing in the genetic engineering to obtain the product. Next question is the restriction endonuclease are used in genetic engineering. Endonuclease, now see, endonuclease, restriction endonuclease, that is known as the molecular is in the genetic engineering. Because why we are using these enzymes? Why we are using this enzyme? Because they cut DNA at a specific base sequence. Means one endonuclease cut both, either they are the foreign or they are the host, they will cut at the same base pair. Next is here a genetically manipulated organism. Genetically manipulated organism means already recombinant DNA or contain its genome one or more inserted gene of the another species. Means एक एनिमल्स जो एक एनिमल है या ऑर्गेनिज्म है जो हम बनाए हैं, कैसे बनाए हैं? जेनेटिकली मैनिपुलेट करके बनाए हैं. उनको अपन क्या कहते हैं? दैट इज नोन एज द ट्रांसजेनिक ऑर्गेनिज्म. Whether it is having one or more than one gene, means more than one means more than the foreign gene. So that animals are known as the transgenic animals. Next here, this question give you a more enough idea. Now read this question very carefully. What this question stand for? This question says this question says that the basic procedure. Here the procedure is given. The basic procedure involved in the synthesis of recombinant DNA. Which DNA? Recombinant DNA molecules is depicted below. Here this diagram is given. The mistake in the procedure is, what do you have to find? You have to find that what is the mistake. What is the mistake in this procedure is done. So let us see. Here you are having one bacterial. This is the plasmid. This is the bacterial chromosome. This is the plasmid, whatsoever it is. Here you are having a DNA that is written here. What is given? They have given mammalian DNA. They have given. Now here it is given the cut with restriction endonuclease 1, and here it is given cut with the restriction endonuclease 2. Now you have find it out. You have catch, I think, that here the endonuclease 2 is given and here the endonuclease 1. This is only the fault that this DNA will be not able to become the restriction recombinant DNA because we have used the different type of restriction endonuclease. So that is why what mistake they have done. So there is a many option. To the option there is given two different restriction enzymes are used. We should remember that in when we are using to cut the DNA of the host or any foreign gene or alien gene or gene of interest, so we should use only one type of restriction enzyme. Okay. Now here, next question you people are having here, that restriction endonuclease are used in genetic engineering because now see that they are here clarifying about because why we are using it. Why we are using it because they can cut the DNA at specific base. They are having the design. They are having the very good pattern to cut the DNA. They can cut the DNA at the specific base. Now here, when the genotype of an organism is improved by the addition of foreign gene, the process is called what? So here genotype organism is improved by addition of foreign gene again. So this is known as the genetic engineering. Okay. Then genetic manipulated, the same question is repeated here. That is known as the transgenic. Means again and again they will ask you the question in the one or other way. You have to be very clear about it. Now here, which vector is commonly used in the transfer of gene in the crop plant? So crop plant, to which vector is always used? From which vector it is used? So that is the Ti plasmid of agrobacterium. That is what Ti plasmid of agrobacterium. This name you just know very well. Okay. Now here the tumor inducing. This is the Ti. Ti word. This Ti word is the tumor inducing the capacity of agrobacterium. That is tumefacin. Agrobacterium tumefacin is the two name. Is located in the large extra chromosomal plasmid, and it is called what? So it is short name is what? It is called Ti plasmid. So here Ti indicates what? Tumor inducing plasmid. Tumor inducing. Next we are having here chimeric DNA is which DNA is known as the chimeric, that is recombinant DNA. Now genetic engineering aims at what is the aim, what is the principle, what is the main motto of this genetic engineering is curing the animal disease by introducing new gene. If it is required like ADA disease, if you know, so that can be cured by the implantation of the gene that we will discuss in further slides. Okay, further question it is coming over there. So curing the disease by the introducing new gene. When we will discuss about the applications, so very well we are just going to discuss the question I have made topic wise. So we will at least we will find the maximum question from each topic. So here the genetic engineering aim at the curing what is that curing human disease by introducing the new gene. Next is what the piece of nucleic acid. The piece of a nucleic acid using to find out a gene by forming the hybrid with it. It is called what? DNA proof. Means the piece what you are having that is the DNA proof. जो हमारे पास एक पीस है एक डीएनए का टुकड़ा है तो उसको क्या कहते हैं अपन दैट इज नोन एज व्हाट डीएनए प्रोब इट इज नोन then here it is known as the tag polymerase you see this tag polymerase okay tag polymerase which is used for the amplification of DNA related to which technique so again here okay Thermus aquaticus. So here the polymerase, this enzyme is obtained from the Thermus aquaticus. So now here amplification of the DNA is by PCR technique में हम उसको यूज करते हैं कहाँ पे यूज करते हैं अपन इसको PCR टेक्निक में अपन यूज करते हैं now here the plasmid has been used as a vector because the plasmid vector क्यों यूज करते हैं because it is circular DNA which have the capacity to join to the eukaryotic DNA means here it is the circular DNA and it is having the capacity to join the DNA. It can be get joined over. It is cut from here. It can be joined to the DNA. Now, what is true about the plasmid? See, everybody is having its characteristic. Everything is having its own characteristic. So what is that? Is why the true plasmid. What we call about the true plasmid? So it is that plasmid is widely used in the gene transfer. Where it is used? Okay. So plasmid are widely used in a gene transfer. Now, which of the following cuts the DNA from a specific place? So that is again restriction endonuclease. Restriction endonuclease cut it the DNA from the specific place. This thing you remember DNA restriction when we study about the restriction endonuclease, so it cuts from the specific place. Then here the manipulation of the DNA in the genetic engineering become possible. What is that? Manipulation. You can do manipulate means you can alter the DNA, cut here, join again, that you add over here and make the something else. So that is known as with the help of what? Restriction endonuclease. If these restriction enzymes are not been has not been discovered, it is not it is not possible. Now, which one of the following has the found the extensive use of genetic engineering work in the plant? Again, the agrobacterium tumefacin. This is always used in the main. It is used in the plant. Remember it. Now, restriction enzyme. So their characteristic. What are the characteristics of restriction enzyme? One of it is given that are endonuclease. One thing which cleave the DNA at a specific site. तो क्या करता है? DNA को एक specific site पे cleave करता है. Suppose DNA A है, DNA B है, तो एक specific place से ही cut करेंगे this endonuclease. Now here restriction enzyme, restriction endonuclease. What happens in the case of prokaryotes? So why it is that they are synthesized by the bacteria as a part of their defense mechanism. They use as a defense mechanism. Restriction endonuclease in bacteria in prokaryotes they are used as a restriction or defense mechanism. The TI plasmid is often used for making transgenic plant. This plasmid is found in. Now, what is that? Tumor-inducing plasmid that where they are found? Agrobacterium. They are the part of what? Agrobacterium. Now, again, it is asking that which of the following example to direct gene transfer? You want to transfer the gene, direct, direct gene transfer. You have to transfer the gene. You will use what? Microinjection, electroporation, particle gun, or ballistic gun. What we call it is a Now, how many copies of DNA sample are produced by PCR technique in cell cycle? Here it is given the 64. Now, thermal cycle, okay, thermal cycle take place in which technique? So again, here it is that it is take place in the PCR. This is the quick revision. The RDNA technology, which is the following technique, is not used in the conjugation. PCR technique is used in the production of genetically modified food. PAC and YAC is what? Artificial DNA obtained from the bacteria. So, friends, for today's for this much, and there are lot of questionnaires I have prepared chapter wise, and you will get and the unique question which is very important questions which are always asking in your exams. So be ready and subscribe these channels to get